Hi everyone, in this video I am going to explain you about the remaining part of cell wall and cell membrane. Pits. Unthickened areas or depressions in the secondary cell wall are called pits. Pits of the adjacent cells generally occur exactly opposite and form functional pit pairs. The cavity unthickened area of secondary wall is called pit chamber or pit cavity. The opening of the pit cavity into the cell is called pit aperture. The membrane, middle lamellum, which separate the pit pair is known as pit membrane. Pit membrane is permeable. Pits are useful in intercellular transportation. Based on the shape of pit chamber, pits are classified into two types. Number 1, simple pits. Number 2, bordered pits. Number 1, simple pits. The diameter of the pit chamber is uniform because of the absence of border. The diameter of the pit aperture is equal to the diameter of the pit cavity. The thickness of the pit chamber is also uniform. It is represented by single circular in cross section. Let's check the diagram carefully for better understanding of these simple pits. Bordered pits. The secondary wall overarches the pit chamber and forms border all around the pit. The diameter of the pit cavity is not uniform. Due to the presence of border, the diameter decreases centripetally. The diameter of pit aperture is smaller than pit cavity. The pit membrane bears centrally located lens-shaped thickening called torus. The thin elastic membrane surrounding the torus is called margo. Torus is useful to prevent the rupture of pit membrane due to unequal pressures. It is represented by circle within a circle in cross section. Let's check the diagrammatic representation of bordered pits carefully. Cell membrane. Cell membrane is also known as plasmalemma or plasma membrane. The term plasmalemma was coined by Plowe. The term cell membrane was coined by Nageli and Cramer. Chemical studies on human red blood cells (RBC) enabled the scientists to deduce the possible structure of plasma membrane. The detailed structure of cell membrane was studied only after the advent of electron microscope. The phospholipid layer is sandwiched between the two protein layers. According to this model, it is solid in nature and it cannot explain the selectively permeable nature of cell membrane or plasma membrane. The cell membrane is thin, elastic and selectively or differentially permeable. It is found between protoplasm and cell wall. It can be seen under the microscope in a plasmolysed cell. The thickness of the cell membrane is 75 angstroms. It is made up of proteins, phospholipids and carbohydrates. The lipid content of the membrane is mainly phosphoglycerides. In human erythrocyte, the composition is protein 52%, lipid 40%, carbohydrate 2 to 5% in RBC or erythrocyte of human cell. The phospholipids are also called amphipathetic in nature, which means the phospholipid molecules contains both hydrophilic and hydrophobic parts. The lipids are arranged within the membrane with the polar hydrophilic head towards the outer sides and the hydrophobic tails towards inner side. This ensures that the nonpolar tail of saturated hydrocarbons is protected from the aqueous environment. Carbohydrates of the cell membrane are oligosaccharides, which are branched or unbranched. Proteins present in the plasma membrane are three types. Structural proteins, which are backbone of the cell membrane. Carrier proteins, which are transport materials. Catalytic proteins, which are enzymes. Structure of the cell membrane. Various models of the cell membrane were proposed by different scientists. Number 1, trilamellar or sandwich model or PLLP model. It was proposed by J. Danieli and H. Davison. in the year 1935. According to this, cell membrane consists of a bimolecular layer of lipids which lies between two layers of proteins. The polar hydrophilic heads of each phospholipid layer are towards the surface, and non-polar hydrophobic tails lie together in the central region of the membrane. The phospholipid layer is sandwiched between the two protein layers. According to this model, it is solid in nature and it cannot explain the selectively permeable nature of cell membrane. Unit membrane concept. It was proposed by the scientist Robertson in the year 1957. He found that all biological membranes are made up of lipid layer sandwiched by two protein layers. It is similar to trilamellar model. The unit membrane has the thickness of 75 angstroms. This model could not explain the dynamic and functional nature of plasma membrane. This concept is not valid because different biomembranes differ in their form, composition, and thickness. Fluid mosaic model. It is the most accepted model of plasma membrane. It was proposed by S.I. Singer and G. Nicholson in the year 1972. According to this model, cell membrane is semi-fluid in nature. The lipids form the liquid matrix which is impermeable to water, and those are water soluble compounds. Protein molecules are arranged as mosaic pattern in the lipid matrix. These protein molecules resembles floating icebergs in lipid ocean. Depending on the ease of extraction of membrane, proteins are two types. Number 1, extrinsic or peripheral proteins. These are present superficially on the outer and inner surface of the membrane. They are easily removable. Example, Spectrin, Cytochrome. Second one, intrinsic or integral proteins. These proteins run through the lipid bilayer and known as tunnel proteins. They provide channels for passage of water and water-soluble products. They cannot be removed easily. Example, carrier proteins. Let's see the diagrammatic representation of fluid mosaic model of plasma membrane. Check this diagram carefully. How integral proteins are arranged, how peripheral proteins are arranged, how sugar molecules are present, how phospholipid bilayer is laid in the diagram. Let's check this diagram carefully for your better understanding. Up to here, the cell membrane is completed. In next video, I will explain you about another cell organelle of eukaryotic cell. Thank you.